Anastasiia.
Hello, everyone. I would like to also thank organizers for inviting me today so that I can present my talk titled Hidden Impact of Maternal Contribution Variability. And with that, I would like actually to start with the definition of variation, despite the fact we've heard of it today a lot of times. It's basically a difference between cells, organisms, or group of organisms caused either by genetic differences or by the effect of environmental factors. And there are a lot of sources and mechanisms involved in variation, and they can be grouped into genetics, of course, environmental effects, and also epigenetics. And they all interplay with each other to result in a final phenotype we can observe and measure. Why is it important? Because first of all, variation contributes a lot to the evolution of the species, their diversity, and last but not least, to their adaptation to changing environmental conditions. It's important to distinguish between variation and noise. It's something we are talking a lot today. And of course, variation is not equal technical noise, which is something we are very familiar with. But also, it's not often equal biological noise, which is stochastic fluctuations in gene expression and often unpredictable. But it's important to know that this stochasticity in gene expression can generate phenotypic variation, but it's not the only source of variability in gene expression. With those being said, I would like to introduce you to maternal RNA. Maternal RNA is basically maternally provided RNA deposited into the egg by the mother. And why is it so important? Because first of all, it regulates and directs early cell plate specification and development. And as you may see on your right, maternal RNA is actually impacting and directing cell plate specification in majority of vertebrates, and is active before zygotic genome activation takes place, which, for example, in zebrafish can take up to 6 cleavage cycles and 3 hours post-fertilization. And it's also important to know that maternal RNA is the only source of information at the beginning for the newly forming organism. It's also very stable because there is no transcription, translation, or RNA decay within egg. It's basically a snapshot of information provided. So when fertilization event takes place, the newly formed organism is ready and can start developing. It contributes to non-genetic inheritance and integrates information experienced by the mother. And to follow up on this, I would like also to mention the work of the member of my group, performed on the freely swimming round goby, when they actually sampled them on different summer and spring months and different temperatures of the water. And what they saw that this temperature is actually affecting the maternal composition of the egg, leading to their diversification over time. But there are still open questions remain. First of all, is maternal RNA variable under normal conditions? And what maternal RNAs are actually variable under normal conditions? And also, how do specific stimuli contribute to this variability within the population? Does it increase the bed hatching, basically increasing the survival rates of the population? With this, I want to introduce you to our project. It might look a little bit complicated right from the start, so let me guide you through it. So what we actually did, we did a heat shock of our population for 2 weeks at elevated temperature. And then we performed the 3 consecutive samplings of our fish immediately after heat shock, 2 weeks recovery at a normal temperature, and then 1 month's recovery in a normal temperature. We collected from all the time points unfertilized oocytes for RNA sequencing, and also collected offsprings for further behavioral analysis. And also to check for epigenetic inheritance and transfer to the next generation, we did the same for F2 generation. For the focus of today's talk, I will focus only on F1 generation unfertilized oocytes. I want to additionally stop on a sample collection procedure because it's really important. We picked at least 6 fish per group and at least 20 unfertilized oocytes per female. They were all gently picked one by one with tweezers and placed into DNA RNA shield to preserve the content of the cell. But as we noticed, the DNA RNA shield is actually hardening the chorion, not maybe penetrating the cell properly. We also applied the crushing of the oocyte inside the DNA RNA shield. Then it was triazole-based RNA extraction and total RNA sequencing of our samples. In the end, we needed, of course, to filter out so-called technical outliers, resulting in the final number of cells and oocytes left. And as you may see in here, there are sample names is basically a mother individual highlighted. You see that the number of oocytes is much more lower than the initial numbers we used, highlighting an importance of using a lot of replicates for this type of experiment. And with this in mind, please, I hope you can appreciate that we can see the mother-specific signatures in our data. All the mothers are highlighted in different colors, and the samples derived from the same mother are tending to cluster together. We also see some group-specific patterns, basically control being more clustered on the left side of the graph, and treatment being more spread and variant across the PCA1 plot. And what also is important to mention in here is that we do see a heatshock effect in our group, in our treatment group, with highlighted heatshock proteins being upregulated in the treatment group. Okay, so we saw the variability. How can we actually measure it in the data? And unfortunately, there is no unified way to do it in the literature. I did a summary of a few things used. Of course, mean median is not a variability measure, but it's something that is important to keep in your data for further reference. Standard deviation variance is something that shows you more or less the same information, and also they are biased towards highly expressed genes. Fun factor and coefficient of variation is something that was created to overcome this issue, but they appear to be biased over genes with low expression. And last but not least are median absolute deviation and interquartile range, which are not sensitive to outliers, kind of, but at the same time, it's because they are focusing only on the central distribution of your data, basically ignoring the tails. If you have any other thoughts or am I missing something, please get in touch. But as for now, we are focusing only on the variance as a measurement of variability in our data. So as I mentioned before, variance really bias towards the genes with higher expression. And as you may see here, the higher the mean expression, the higher the variance. The way to overcome this issue is, of course, the variance stabilizing transformation of your data, which removes this technical dependence and allows you to see a much more cleaner, hopefully true biological meaning of your data. And if you go for a closer look now on your data set, and here you see a random example of a gene, and you see that, for example, a group variance in here is pretty much high, and it might be driven by individual mothers, which is good, that's what is expected. But what is also interesting to see is that you see a specific individual differences. For example, in here and the mother D8, you see that there is a very high intra-individual variance of all sites derived from the same mother. So it's also interesting and important to keep track of potential intra-individual differences in our case as well. So how can we actually do that? The way we propose is to do a ranking of your genes. And the idea behind is basically to assign the genes to the deciles from 1 to 10, with 1 having the 10% of the genes with the lowest variance across their gene set, and the 10th decile having the genes with the highest variance. In the end, you receive something like this. You see a list of genes, and they all have the deciles assigned for specific mothers. It looks already pretty much complicated, especially if you have 20+ thousands of genes. So how can you actually overcome this? Is to assign categories. In here, what we propose is, for example, to assign the category as consistently high variance, where you know that the gene is constantly within the 10th deciles across all the mothers. You can go with individually driven high variance when the gene is still within the last deciles, but it's variable across all the mothers. And the same principle can be applied to the low variance and the medium variance. And if we'll focus only on the high variant genes, you can clearly see that there is a small portion of genes which is shared between treatment and control group, with a group-specific pattern still being present. And if we'll focus on the function of the genes involved, by checking geoterms. You can see that in control group, there are not so many geoterms yielded, but at the same time, they are involved in developmental process or regulations. And if you'll do the same for the treatment group, you can already hopefully appreciate that there are much more geoterms involved. And at the same time, they are involved in very different processes. So for example, in here, you can see that they are clearly involved in some translation mechanisms and processes, and also that there are some metabolic processes involved, which is kind of good, also confirming that our heatshock worked. With thus being said, I have a take-home message for all of you that parental heat stress can lead to measurable diversification between oocytes and zebrafish. There are a few conclusions we can actually make that maternal RNA is a very promising molecule to understand the aspect of variation, and also our findings suggest that maternal RNA can mediate heritable diversification in evolutionary processes. What we still need to do is to finalize the pipeline to analyze variability, and we also need to assess the persistence of the treatment effect over time. As you remember, we had like 3 different time points, and also over generations. And with that being said, I would like to go to acknowledgements and specifically highlight our research group, which participated in making this project. Also, Swiss National Science Foundation, which made this project possible, and other groups involved and helping us to fine-tune it. Thank you for your attention so much, and I'm ready for questions.